Thank you. I couldn't be more excited than standing here in front of this relevant and engaged and inspiring audience to share with you what we've been working on at Karius for the past 3 years or so, and why I think that genomics is going to completely change the field of infectious diseases. Let me start with a story of a 5-year-old ALL patient that checks into a hospital not far away from here with fever and neutropenia. Blood cultures return positive for Klebsiella pneumoniae, and they put her on cefepime, which seems to do the trick, and the fevers go away. A few days later, the fevers are back, and cefepime is no longer effective. So they take her through a lot of blood works and testing, including blood cultures that come back negative, including fungal cultures negative, including a couple of PCR panels for pathogens, GI pathogens, and a couple of viruses, including biomarkers for fungal diseases, fungal infections, and a few other serology testing. All of those come back negative. They take her for CT scan, which reveals those nodules in the lung, suggesting an infectious process. They cannot biopsy her because she's too weak to survive a biopsy. And here we are standing and looking at this information not being able to help that patient. And this is first world, 21st century, best possible institutions, best clinicians, top tools, top technologies, and we cannot help this kid. Why is this broken? Let me suggest that this chain between being healthy, sick, diagnosed, treated, and back to healthy is broken at the diagnosis stage. And the reason it's broken is because we just don't have the tools right now. If only we knew what that patient has, there's a very high likelihood that we could treat her. We spent a lot of energy as a society, a lot of effort to devise therapies for those specific microbes. The problem is just that we do not know what we're going after. And that is the problem here. Obviously, this is not confined to the US. Infectious disease is a big problem worldwide. It's actually the second leading cause of death worldwide, second only to heart disease. So more people die of infections than of all cancers combined every year. It accounts for about 16% of global mortality and 2/3 of death in children under 5 years old. And while heart disease and cancer have a steady hit rate on humanity, infectious disease has this property that it can suddenly spike in the form of a pandemic and have a much higher impact than even those numbers here. And to make it even worse, we humans devised some therapies and we're overusing them, thus helping those microbes be even more resistant and become a bigger problem. And the reason we're overtreating is, again, back to the inability to diagnose and throwing the book at those patients and trying to see what works for them. So in an attempt to try and quantify how big this problem is, we started a study with Stanford last year in which we enrolled 350 patients that presented to the ER with sepsis. And we followed each and every one of those patients a long time to check when is it that this patient is diagnosed. And the picture that emerges is pretty grim. Even after 8 days, only 38% of those patients are diagnosed. At day 2, about 18% of the patients are diagnosed using blood cultures. And then it's a long and tedious investigation and grind up with additional cultures and serology tests and PCRs and one-offs to get to this 38% figure. Now, the picture is not as grim as it may look because it's known that about 40 to 50% of septic patients actually are not septic because of an infection, but other causes. But still, what we can see here, what this tells us, is the story of lost time, lost money, irrelevant drugs, confusion, suffering, and mortality. And so the question is, can we do better? Maybe genomics can help us here. And the answer is yes. And specifically, cell-free DNA has been shown to be a very informative analyte in a majority or in a set of other diseases, starting from NIPT, basically completely displacing the need for invasive amnio procedures with a simple blood draw. Very nice results on transplant rejection and the ability to predict a rejection using the donor to recipient cell-free DNA ratios. A lot of interest and a lot of applications and great results in the clinic on circulating tumor DNA. Actually, a whole session in this meeting was dedicated to that topic. And it was actually while I was in the lab of Steve Quake, who's here in the audience and will speak in half an hour or so, while I was in the lab at Stanford, we were working on a transplant rejection research and found something very interesting. We found a weak signal coming from microbes. There's good news and there's bad news. The good news is that if acquired, this signal actually spans bacteria, viruses, fungal infections in the patient. The bad news is that this precious signal is buried in about 1 million-fold more human DNA. So the signal-to-ratio problem here is immense. It's about 100,000 to 1 million-fold signal-to-noise. And that's in stark contrast to other applications. So orders of magnitude lower signal even than oncology applications. And I think those figures tell us maybe why this has not been looked at. This has not been looked at previously, and certainly why that has not been used previously for diagnostic applications. So how does that cell-free DNA from microbes get into the blood? This cartoon shows an infection in the lung, and as those microbes are dying and shedding their DNA, the DNA is absorbed into the plasma, which is where we pick it up. We then separate out the human DNA and sequence the microbial DNA and use this genetic information in conjunction with a big, large database of tens of thousands of microbes to map the reads and figure out which microbe or combination of microbes are accounted in that tube of blood. And we return our clinical result based on that information. Now, it turns out that cell-free DNA is very, very useful for infectious diseases for 3 reasons. It can tell you everything from everywhere in real time. The everything has to do with the fact that all those classes of microbes, bacteria, viruses, nematodes, protozoa, fungi, they all have a DNA or an RNA genome somewhere within their cell, and they all shed it as they replicate and die. And so with one assay looking for this genetic material, you can basically cover all of those very different classes. The everywhere aspect comes from the fact that every cell in our body is touched by the blood system, and then cell-free DNA there is aggregating the data and combining the data from everywhere in the body, which allows us to see not only blood infections, but also deep-seated infections in lung and heart and brain and abdomen, because they all are touched by the bloodstream and they all are shedding into the bloodstream. And the last aspect is the real-time aspect. In this cartoon, the red signifies what is the microbial load, and you will notice that the cell-free DNA has the potential to follow it very, very closely, because the half-life of cell-free DNA is about 30 minutes, which means that if we see a fragment of DNA, it means that there was a microbe replicating in the body and shedding its DNA very, very recently. This is, again, in contrast to cultures that become sterile as soon as antibiotics are administered, and serology, which remains on forever long after the infection is actually cleared. So this everything, everywhere, in real time property of cell-free DNA makes it a very interesting analyte for infectious disease diagnostics, providing a very broad and sensitive way to measure. So what's not to like? There are a wide range of challenges when dealing with such a diverse set of microbes with different biologies. Is one of them. The GC content of those microbes is very, very different. So the assay needs to be able to capture in a quantitative way, in a very accurate way, across 25 to 80 GC content. the genome length of those microbes is also very different. And so again, trying to capture everything between a few kilobases to hundreds of megabases has its own challenges. Those microbes, unfortunately, that infect patients do not resemble 100% what was deposited and what is measured in publicly available databases. And so they evolve away, and how that connection between what's out in the community and what is in the databases affects your results. And last but not least, the challenge of environmental contamination. So our tubes, everybody's pipettes, reagents, sequencing reagents are full of microbial DNA. Those enzymes are grown in microbes, and while there's no live enzyme in your tube, there's a lot of microbial DNA. There's a whole zoo of microbe DNA there. And if not correctly accounted for, one would end up reporting the reagentome as opposed to the infectome of the patient. And which has been, by the way, the case in some interesting papers that ended up being retracted because they identified that it's actually some DNA extraction kit that provided this signal. So lots of very, very interesting challenges. I could probably stand here for the remainder of the day and just tell you how we solved those. We're putting it all into a publication together with a very interesting analytical validation approach that we took, and this will soon be available. And also, I'm happy to talk to anyone today, this afternoon, and provide a little bit more of this exciting science behind the scenes. But once taken together, we were able to devise an end-to-end solution which takes blood as an input, goes through our proprietary sample processing, sequencing using Illumina sequencers, analytics, and returning a clinical result, and all of that within 1 day and across 1,000 pathogens. So with this framework of a CLIA and CAP accredited lab and with a fast turnaround time, let's see what we can do for those septic patients. So what I did not tell you is that at the time we enrolled those patients, we took a blood draw from each and every patient at the time of admission. Shipping the sample would take 1 day, and that's 1 day of lost time. Analyzing it is another day. And the question is, what would that look like using genomics? And the results are pretty stunning. We get on day 2 to about 50% diagnosis of those patients. So this area there between those two lines here, this is all the lost money, lost time, irrelevant drugs, confusion, suffering that we can avoid using genomics. So this now starts to look more like modern medicine than this slow grind towards a diagnosis at day 8. Let's go back to the 5-year-old patient that we kept aside for a second. Luckily, at the time she was admitted, that institution started using our technology, and after this CT scan, a tube of blood was sent to us. We received the tube. Within a day, we returned the result, which was human HHV6, not surprisingly, but importantly, Rhizopus. So Rhizopus is a mold that is known to infect immunocompromised patients. The problem is that the biomarkers for fungal infections come back negative. The problem is that the first line of defense and the antifungals do not work against that mold. Luckily, there's a specific, well-targeted medicine for that. They put her on that targeted therapy, and the girl recovered. What that demonstrates is not only the breadth of the test, but also the ability to get to deep-seated infections without the need for a biopsy. And we call this mode of using genomics in infectious disease liquid biopsy for infectious diseases. Basically, very similar to how liquid biopsy for oncology has obviated the need for invasive biopsies. In this study, what we show across those 11 patients, the results from an invasive biopsy compared to the result of a blood draw taken right before those patients went into the OR. And you can see a very nice correlation and actually agreement between those two across bacteria, viruses, a couple of TB cases here, very broad and very accurate. So we have now the technology, because we all together as a community developed some of those protocols, the sequencing, the analytics. We've wrapped it up into a CLIA and CAP accredited solution, and we can help those patients. We can save the time, the money, the suffering, the confusion. We can save the biopsies. So we're done, right? Absolutely not. Actually, this is just the first piece of the journey, and I think that the next phase is going to be even harder. As we take it from helping 350 patients and 1,000 and 10,000 patients to really becoming the paradigm in infectious diseases. And it's only by working together with tool manufacturers and regulatory and reimbursement and academics and clinicians that we will be able to take that such that we can touch as many patients as quickly as possible, but also being very cognizant of the fact that medicine is slow to change, and for a good reason. Patients' lives are at stake, and so we need to move as quickly as we can while maintaining patients' safety. So if you want to have a closer look at what we have been doing, there's a lot of publications and talks and interviews on our website. Happy to help your patients through our CLIA and CAP accredited lab that is now operational in Redwood City. We would love to collaborate on more clinical studies. The more, the better. Different use cases. Karius X is our moonshot project. And that's where if you have a high-risk, high-reward project that you think could benefit from this ability to probe at high resolution the blood infectome or the blood microbiome of patients, we would love to hear about that. And if you want to have an even deeper look, we're obviously recruiting engineers, bioinformaticians, molecular biologists, clinicians. So I'd like to thank you on behalf of the entire Karius team for being such an engaged community. Some of our collaborators, some of our customers are here in the audience. We all look forward to an exciting but not easy next piece of this journey as we take this approach and make sure that it touches the millions of patients that it has to touch. Thank you very much.